Hello dear viewers, welcome to Mr.Zoologist YouTube channel. Today we are going to discuss about cytoskeleton in detail. Cyto means cell, skeleton means structure. So cytoskeleton is an organelle. These are used to maintain the shape and structure of the cell. So in this diagram we are seeing is a scan of the first electron micrograph taken intact of eukaryotic cell in which cytoskeleton is holding the shape of the cell. So, cytoskeleton is the structure consisting of fibrous proteins that occur in the cytoplasm and maintains the shape of the cell. So, the eukaryotic cell is a 3D structure. It has a cytoskeleton anchored to proteins in the plasma membrane. These proteins both maintain shape and allow movement. The cytoskeleton is the dynamic structure as the microfilaments and microtubules. They have the ability to depolarize or repolymerize very easily. Means to say they can lose their shape or they can gain their shape. They can grow as well as they can be destroyed according to the demand of the cell. So if we talk about the structure of the cytoskeleton, it is a network of filamentous proteins. Filaments formed from a few proteins, monomers proteins forms polymer filaments. Means to say smaller units combine together to form the larger networks of the structure of the cytoskeleton. So if we talk about the location of the cytoskeleton, it is located in the nucleus and cytoplasmic compartments. So actually, if we talk about its exact location, not within the organelles, but they are present in all other structure of the cell, including the cytoplasm and including the nucleus. If we talk about the location based upon cellular function, they are named on the basis of the physical size of which they have. So if we talk about the functions, interactions or functions based upon the filament physical properties, such as what is the strength of the filament, what is internal strength, and how does it give shape to the cell, and how does they provide the motility for the movement of the cell, like inside the cell movement, whole cell movement, or motor proteins associated with two filament systems. So they are also involved in the signal transduction, transport of the signals from one part of the cell to the other part. So if we talk about the extracellular matrix, that has a similar structural role outside the cell as cytoskeleton has inside the cell. The same function is performed by the extracellular matrix, which is present on the outside environment or the extracellular environment. So there are three types of the cytoskeletal filaments. First of all is the microfilaments, second one is the microtubules, and third one are the intermediate filaments. We are going to look upon these three cytoskeletal filaments in today's detailed lecture. So keep attached with the video and don't forget to like and subscribe our video as well. So if we talk about intermediate filaments first, they are different, present in different cell types, and they are different intermediate filaments. All eukaryotes, a nuclear cytoskeleton is the same. They resist the stress applied externally to the cell cytoplasm. And if we talk about their diameter, they are approximately 10 nanometer in diameter. And their size is intermediate between microtubules and microfilaments. Cross-linking proteins allow interactions with other cytoskeletal networks. It means to say intermediate filaments are not present alone, they are associated with other cytoskeletal filaments as well. Intermediate filaments associated proteins. If we talk about the IFAPS, intermediate filament associated proteins, they coordinate actions between intermediate filaments and other cytoskeletal elements and organelles. And if we talk about human disorders, their mutations weaken the structural framework, increases the risk of cell rupture. And intermediate filaments have large tensile strength. Their main function is to help the cells withstand mechanical stress. As you can see in the diagram that here, if it apply the mechanical stress, the cell which has the intermediate filaments remains intact a little bit in the bend shape, whereas the cell which do not have the intermediate filaments, they are shown in the bends, fully bent form right here, as you can see in this diagram. Stretching will lead to the deformation of the cell. So next are the intermediate filaments. Form strong rope-like proteins assemblies. If we talk about its structure, that is somehow on the one end is the amino group, and on the other hand is of the carboxyl group or carboxyl group, and they form a rope-like structure, like one twist upon the other, and they form intermediate filaments. And if we talk about in detail about the structure of intermediate filaments, if we talk about its protein composition, intermediate filaments are a diverse family of proteins structured by or encoded by more than 70 different genes in humans. Based on sequence homology and cell type expression, they are grouped into six classes. Type 1 and type 2, they are keratins, they are present in the epithelial cells such as hair and nails. Type 3, Vimentin, Desmin, GFAP, muscle, and they are present in the muscle glial cells and mesenchymal cells. And type 4 are neurofilaments, which are the part of the neurons and nervous system. And type 5 are the lamins, which are present in the nucleus, like nuclear lamina. And type 6 are the nestin, which are neural stem cells, like that which are present in the stem cells, such as zygote, which has the ability to differentiate into multiple functional structures of the cell. And if we talk about the domain organization, that how IFS or intermediate filaments domain looks like, they are made up of central alpha helical rod domain that is approximately 310 to 350 amino acids forms coiled dimers. Dimers means say one end of amino group and the other end is of the carboxyl group, as you have seen in the previous diagrams. If we talk about the variable and terminal head domain regulates filament assembly. They have a C-terminal tail domain which provides structural diversity and regulate interactions with other proteins. That is how a one monomer, one is the head that is consisting of amino group, and the tail that might be consisting of carboxyl group. And on the other hand, you can see that the two monomers are fused around each other and form a dimer. Di means two monomers, and tetramer forms two dimers attached, and four total monomers are in the tetramer. And then they will and the eight will form the unit filament length, and one filament will be equal to the one intermediate filament or complete structure, as you can see in the diagram. Here all type of type 1, 2, type 3, type 4, type 5 and type 6, all types are present in the filament. If we talk about the assembly process or non-polar or ATP to GTP independent working, monomer forms dimer, two monomers form a coiled dimer, and dimers forms tetramer, two dimers associate in an anti-parallel staggered manner like non-polar ends. They do not have the charged ends. If we talk about tetramer forms about the protofilaments, laterally associate tetramers to form the protofilament, and protofilaments further forms the twisted structures, that is rope-like filament that is approximately 10 nanometer in diameter. And that is how the symmetry of the intermediate filaments forms. First of all, monomer transforms into dimer transforms into tetramer into protofilament, and protofilament finally forms a intermediate filament or IF that is approximately 10 nanometer in diameter. If we talk about the functions of the intermediate filaments, they provide the mechanical support, provide tensile strength to resist stretching and mechanical stress in epithelial cells. Keratin filaments connect to desmosomes and hemidesmosomes, stabilizing tissues under mechanical strain. Strain, you might be familiar with it. Strain is actually the deformation in the original shape after the shear stress is applied. If the shape returns to its original length, then it has the elasticity limit not breached, that is intact. If its shape destroys after applying shear stress, that means to say the body has lost its original shape. In neurons, neurofilaments maintain axonal diameter and thus regulate conduction velocity. That's conduction velocity is of the transmission of the nerve impulse or neuron from one neuron to the other neuron. If we talk about organelle positioning and integrity, Vimentin networks stabilize organelles such as mitochondria, endoplasmic reticulum, Golgi bodies, nuclear lamina forms the nuclear lamina, providing structural support to the nuclear envelope and regulating chromatin organization. If we talk about the signal transduction and regulation, IFS interact with signaling proteins, kinases, phosphatases, scaffolding proteins, modulating cell cycle progression. It means to say cell cycle progression is associated with the assembly and disassembly of the different structures associated with the mitotic apparatus and cellular organization or providing shape to the cell. And they are also involved in the apoptosis process, which is a programmed cell death and stress response. They are also involved in the phosphorylation of IFS that controls their assembly and disassembly, such as the nuclear lamina disassembled during mitosis, as you might be familiar with the prophase, in which nuclear envelope disintegrates and the genetic material is exposed in the form of the chromosomes. Earlier on, before prophase, chromatin material present, which is decondensed state, expanded state of the genetic material. So cell migration and plasticity is another function of the IFS. Vimentin is essential for mesenchymal cell motility and epithelial to mesenchymal transition, a process important in wound healing and cancer metastasis. Metastasis is the spreading of the cancer. And if we talk about the barrier and defense, keratins protect epithelial cells from chemical and mechanical inserts. Mutations in keratins lead to blistering skin diseases such as epidermolysis bullosa simplex. And if we talk about the another type of the cytoskeleton, that is microtubules. Microtubules are the largest cytoskeletal filaments. They are approximately 25 nanometer in diameter, and they are made up of alpha and beta tubulin heterodimers that assemble into long hollow cylinders. They are polarized, highly dynamic, and essential for intracellular organization, transport, and cell division. And if we talk about the structure of the microtubules, their alpha tubulin and beta tubulin form a heterodimer, the basic building block of the microtubules. Each tubulin binds GTP, but only the beta tubulin subunit hydrolyzes it, while the A subunit is incapable of hydrolyzing GTP. One GTP might be equivalent to the one molecule of the ATP. If we talk about the protofilaments, tubulin dimers assemble head to tail into linear chains to form protofilaments. Certain protofilaments align side by side to form a hollow microtubule. If we talk about the polarity, the plus end is the fast growing, mostly beta tubulin exposed, and minus end is slower growing, usually anchored at microtubule organizing centers like centrosomes, which are present in the cell. Centrosomes are actually responsible for forming the mitotic apparatus. That mitotic apparatus is essential for forming the metaphase plate and separation of the chromosomes during the anaphase. So they also provide dynamic instability. Microtubules constantly undergo polymerization and shrinkage. Polymerization means to say that, they combine with the GTP to form the microtubules, and shrinkage means to say breakage. These GTP binded molecules then releases GDP, and the structure of the microtubules is lost. So that is controlled by the GTP hydrolysis on beta tubulin. So as I told you earlier that only beta tubulin is capable of hydrolyzing GTP. So this behavior allows cells to rapidly remodel their microtubule network. And if we talk about the functions, they are involved in intracellular transport such as tracks for motor proteins. Kinesins move cargo towards the plus end, which is towards the periphery, towards the dark end. And if we could talk about dyneins, they move cargo towards the minus end, which is located near to the centrosome. If we talk about the transport vesicles, they are also acting as a holding structure for the organelles and protein complexes, providing support to them. They also provide cell shape and mechanical support such as rigidity and resist compressive forces. If they are also defining long processes like axons and dendrites in neurons, and they are also involved in cell division, as I told you earlier about the spindle formation and mitotic apparatus, all about that. And they attach kinetochores and apply pulling forces during anaphases. I have told you earlier about that as well. They are also involved in the organelle positioning, like help position endoplasmic reticulum, Golgi, and mitochondria within the cytoplasm. And they also provide motility legs with cilia and flagella. Microtubules form the axoneme, which is a structure composed of 9 doublets plus 2 central singlets. Dynein-driven sliding of doublets produces ciliary flagellar beating. They also provide intercellular signaling, acts as a scaffolds for signaling complexes influencing pathways like cell polarity and migration. So that is the structure of the microtubule. The upper unit is the beta subunit which hydrolyzes GTP, whereas the lower unit is alpha tubulin that is bind with the GDP molecule. That is the structure of the microtubule. Alpha tubulin and beta tubulin are shown. In the red one is the beta tubulin and the blue one is the alpha tubulin, and they form a microtubule that is about top view on protofilaments, and all other units combined together to form a microtubule. That is the microtubules MTS dynamic instability. In the first one is shown that if the rate of the GTP bound or hydrolysis is faster than the rate of the if the rate of the binding with the GTP is faster than the rate of hydrolysis, then the microtubules will grow. It contains a GTP cap. That GTP cap actually adds with the beta tubulin, and the structure grows on until a protofilament transforms into microtubule. And whereas if the rate of GTP hydrolysis is faster than the rate of polymerization, the microtubule will disassemble, and GTP cap is lost as in the form of GDP and alpha tubulin, and the structure is being shrinked. So some functions of microtubules. If we talk about the functions, they participate in a wide variety of cell activities. They are also used to provide ATP to the motors, and they determine the positions of membrane enclosed organelles, and they also are responsible for the migration of chromosomes during mitosis and meiosis. And that is the last one we are going to discuss about today's lecture, that these are microfilaments. Microfilaments are the thinnest filaments of the cytoskeleton, approximately 7 nanometer in diameter, composed mainly of actin. That is a highly conserved protein. They are dynamic and polarized, enabling rapid remodeling of cell shape and motility. And if we talk about the structure of microfilaments, actin monomers, G-actin that binds with the ATP and ADP. Filaments, actin, F-actin, polymerized form of actin arranged into long helical filaments. And if we talk about the polarity, actin filaments are polarized plus barred and fast growing, and that is actually ATP actin preferred. And minus end or pointed end, that is slower growth, that ADP actin preferred structure. This polarity is essential for processes like treadmilling and directional motility. And if we talk about the organization in cells, cortical actin meshwork is formed beneath plasma membrane that provides cell shape and stiffness. And filopodia and lamellopodia, thin and sheet-like protrusions for migration, stress fibers bundles that provide tension, adherence junctions, actin filaments link cells together and to the extracellular matrix. And if we talk about their function, they provide cell shape and mechanical support, maintain cell surface tension and cortex structure, provide scaffolding under the plasma membrane, provide cell motility with the help of filopodia and lamellopodia. Actin treadmilling enables directional movements, and endocytosis, actin filaments regulate vesicle trafficking and membrane dynamics. Intercellular transport is also carried out by microfilaments. Cytokinesis during cell division is also provided by them. They are also involved in the signal transduction, like actin cytoskeleton interacts with the signaling complexes, regulates growth, survival, and adhesion. So that are the structures like they are providing in the cell: the microvilla, small contractile bundles, dynamic protrusions, and the contractile ring. These all are shown in the diagram. So actin filament is formed. Monomer is actually G-actin, and actin filament F-actin is has a polarity due to the plus end and the minus end. And actin filaments bind hydrolyzes ATP like that. If ATP binds with the actin, that will form the structure of the microfilament. And if ADP is released and the structure binds with the ADP actin, that will form the minus end or lower end. That is a pointed end. And filopodium, lamellopodium, and contractile bundles, these are the crawling movements provided to the cell by these microfilaments. These are the movement providing structures in the cells. So they are also involved in the muscle contraction. Actin filaments are anchored to the Z disc at the plus end. The actin filaments face each other in the sarcomere. Sarcomere is the region of the skeletal muscle cell where light band, dark band, and then light band, these contrasting bands are present, and this one region contains all these three different bands all together. Upon contraction, the myosin heads move towards the plus end and pulls the actin filaments towards each other. Like in the first diagram, that central of the myosin head shows that is a A band, that is a dark band. And lower in the lower diagram, you can see that in the contracted state, the actin filaments move over the myosin and they come closer to each other. So if we talk about the function, they provide mechanical strength, link transmembrane proteins, form contractile rings, cytoplasmic streaming is carried out by them, generate locomotion in cells, interact with myosin to provide force for muscular contraction.